Welcome back, everyone, and I hope you are having a great time and enjoying your study sessions. So, today I want to go continue with the biology chapters, and we are on chapter 2. And right now I am doing the assessment questions from Kaplan, and the first question, I have the questions here. So the first question was the correct sequence of the maturation or development of a mature sperm cell. And it is, I got the question wrong, but the correct sequence is first the spermatogonium turning into a primary spermatocyte, and then the secondary one. So I believe until the secondary spermatocyte, it is a haploid, diploid, and then it turns into a haploid. So I drew it here. So spermatogonia and the primary spermatocyte are diploid, and the secondary spermatocyte, and then the spermatid, and last but not last one being the spermatozoan are haploids. And we need to understand where this diploid turns into a haploid. So for that, it actually is directly related to meiosis. So in meiosis, we have the diploid, the 2N, as you can see here. And I believe after, so even for the second question, I believe that one is about the development of an egg cell. So we need to know the development of a maturation of both sperm cell and egg cell. So what part of the life cycle is that? So the stages are, the PMAD 1 and PMAD 2, and for prophase we know that okay, let's actually go through this here. So we expect here, like in telophase 1 or cytokinesis, that it turns haploid. So for the egg cells, from the time of birth to just before ovulation, all egg cells are arrested at the prophase stage of meiosis 1. So prophase 1, it's this one. It's just the crossing over, right? And then so they're arrested here, they stop at here. So when these are primary oocytes, and then at ovulation, the egg cell has completed meiosis 1 and is now arrested in metaphase 2. So we have the chromosomes meeting in the middle. So at ovulation, they stay here before, just separating, right? And you can see here that it is in telophases that the disjunction takes place, basically, right? In anaphase, we have a separation. So the homologous pairs move to opposite poles of the cell. This is for anaphase 1, and this is disjunction, and it counts for the law of segregation. So we have the anaphases here. So anaphase is here and here. So there is a lot of segregation for anaphase 2, spindle fibers contract and separate the sister chromatids, and chromatids, now called chromosomes, move to opposite poles. So these are chromosomes, and these were chromatids, and or chromatids here. And we have the chromosomes decondense and nuclear membrane reforms. So you need to remember that chromosomes are like a single strand, and you have a pair of them, which makes 2 chromosomes, and that's how you have that. Now, in the interphase cycle, which is anything other than meiosis, we have the DNA replication that takes place before mitosis or meiosis, right? That's when you make a replication of the DNA. Okay. Now, metaphase, so the egg cell at ovulation, which is here, the egg cell has completed meiosis 1 and is now arrested in metaphase 2 as a haploid cell called a secondary oocyte. So we have primary oocytes and secondary oocytes. Now, primary oocytes are when a sperm penetrates. So primary oocytes are arrested in prophase 1, and secondary oocytes are arrested at ovulation in metaphase 2. And when a sperm penetrates the outer layers of the secondary oocyte, it completes meiosis 2 to become a mature ovum. Ovum is the singular form, and the plural form is ova, right? Because we have ovaries that have follicles that produce ova. And that is for this question for now. So which of them is correct? From birth to menarche, let's ask GPT, what is menarche? Occurrence of menstruation, which is the beginning of a woman's reproductive years. It is a significant milestone in the physical and emotional development of girls and typically occurs around the age of 12 to 14. So, what is the egg cell maturation before and after menarche? So, from birth to just before ovulation, like the first ovulation, all these egg cells are arrested at the prophase 1, right? The development of egg sites, oocytes. This process begins during fetal development, where a female baby is born with all the eggs she will ever have. Although they are immature at this stage, these immature eggs are called oogonia. During childhood and leading up to menarche, the ovaries continue the process of egg cell maturation. How does... Oh, so until... So all these secondary oocytes at ovulation in metaphase 2, they are haploid cells. But before that, they were diploid cells. So in metaphase 2, right, we have here, yes, in metaphase 2, you can see that so the egg cell has completed meiosis 1 and is now arrested in metaphase 2 as a haploid cell called a secondary oocyte. So a secondary sperm cell is also a haploid. We also have the pictures for them, which is this. So we have spermatogonium, the mitosis happens, its primary spermatocyte is 2N, and then the meiosis happens, and then we have secondary spermatocyte. It is for meiosis where the diploids turn into haploid. However, for mitosis, these are all diploids, right? For mitosis, the cells are diploid and never turn into haploids, right? Mitosis is a type of cell division that produces two daughter cells, each with the same number of chromosomes as the parent cell. As the parent cell, the cells produced by mitosis are diploid, meaning they have the same number of chromosomes as the original cell. In contrast, meiosis is another type of cell division that specifically occurs in reproductive cells, gametes, and results in the production of haploid cells. Haploid cells have half the number of chromosomes as the parent cell. Meiosis is responsible for producing gametes, sperm and egg cells for sexual reproduction. The fusion of haploid sperm and egg cells during fertilization restores the diploid chromosome number in the zygote. So mitosis maintains the diploid number of chromosomes in the daughter cells, while meiosis specifically leads to the formation of haploid cells for sexual reproduction. Some studies suggest that in patients with Alzheimer's disease, there is a defect in the way the spindle apparatus attaches to the kinetochore fibers, at which stage of mitotic division would one first expect to be able to visualize this problem. So, the spindle apparatus attached to the kinetochore sounds like metaphase. Did we get the question right? We put A, prophase. Oh, so I did actually get it right, but I answered it now incorrectly because the spindle apparatus attaching to the kinetochore fibers, that is in like the centrioles going to the two opposite poles, and that is in prophase. In metaphase, that's where they are the chromosomes meet in the middle, and they separate in anaphase. So the spindle apparatus first interacts with the kinetochore fibers near the end of prophase, while the spindle apparatus aligns the chromosomes at the equatorial plate. During metaphase, the initial connection of the microtubule to the kinetochore occurs in prophase. That's pretty detailed. A researcher wishes to incorporate a radio-labeled deoxyadenine into the genome of one of the two daughter cells that would arise as a result of mitosis. What is the latest stage of cellular development during which the radio-labeled deoxyadenine could be added to achieve this result? What is the latest stage? It would be D. I answered C. So we have also this here. So it is S, not G2. So that's when the DNA gets replicated. The latest stage that you can introduce that is the replication. After replication, it's just cell growth. You make the organelles, and you have a checkpoint on G2 for checking the cell size and organelles, and then the mitosis happens. To ensure that the labeled deoxyadenine will be incorporated into the DNA of one of the daughter cells, we have to insert the nucleotide before DNA replication has been completed. Because replication occurs during S stage, we could introduce the deoxy during G1 or S stage. Because G1 precedes S, the latest point at which the deoxyadenine could be added is the S stage. Honestly, G1 still would sound good to me too if I knew how to even answer this, because it's like the latest, it's before the DNA replication. I don't know. Can you do it during DNA replication? Tell me what you think. So next time, if they ask what is the latest stage, and if that stage is also included, so if the DNA is being replicated in S stage, we should answer S stage, not the stage before it, like the latest one. So yeah, certain ovarian tumors called granulosa cell tumors are known to produce excessive levels of estrogen. A physician who diagnoses a granulosa cell tumor should look for a secondary cancer in which of the following parts of the reproductive tract. So, secondary cancer, excessive levels of estrogen, I answered the fallopian tube. A, but the answer is C, endometrium. Estrogen is known to cause growth of the endometrial lining during the follicular phase of the menstrual cycle, and its levels stay high during the luteal phase to promote vascularization and glandularization of this tissue. Excessive levels of estrogen may provide a strong enough signal for cell growth to promote tumor formation or even cancer. The other tissues listed in this question require estrogen for development but are not strongly dependent on estrogen for growth. So, if this is the question, take away only endometrium needs estrogen for growth. The other parts of reproductive tract need estrogen for development. Estrogen is known to cause growth of endometrial lining during follicular phase, not luteal. So you can see here the growth that happens in the follicular phase. And its levels stay high during the luteal phase to promote vascularization and glandularization of this tissue. You want it vascularized and glandularized. And excessive levels of estrogen may provide a strong enough signal for cell growth to promote tumor formation or even cancer. The other tissues listed tumor, cell growth, and we have the endometrial lining. Estrogen. What stimulates the, like, where does this estrogen come from, right? This estrogen is a response to FSH. So you have a moderately high level of FSH, which causes an increase in estrogen. But yeah, the estrogen develops reproductive tract, so vagina, cervix, all the good stuff, like endometrium, cervix, fallopian tube. But it thickens the uterine wall. Yes, the endometrial wall, right? So uterine is uterine the same as endometrial lining? And let's put this question here. I relate to the female, to the uterus, but they refer to different aspects. Uterine refers to anything related to the uterus, which is a hollow muscular organ located in the pelvis. The uterus plays a central role in pregnancy, where it is where a fertilized egg implants and develops into a fetus. The endometrial lining is a specific part of the uterus. Uterus. It is the innermost layer of uterus, and its thickness varies throughout the menstrual cycle in response to hormonal changes. The endometrium is crucial for pregnancy because if a woman becomes pregnant, a fertilized egg zygote implants itself into the endometrial lining and receives nourishment from it as develops into an embryo and then a fetus. So plus endometrium. Endometrial lining is the innermost layer, right? Innermost layer of uterus. Amazing. And but still, where does this estrogen come from, right? That would be here. So let's look at estrogen. We have our gonads. So estrogen and progesterone in ovaries. So it is the ovaries, and testes, testosterone. But where does the estrogen and progesterone come from in males, and vice versa? Where does testosterone come from in females? If we don't have, so where does estrogen and progesterone come from in males, and testosterone in females if men don't have, because men don't have ovaries and women don't have testes. While the primary sex hormones, estrogen, progesterone in females, and testosterone, are often associated with the ovaries and testes, it's important to note that these hormones are also present, albeit in different quantities, in both males. In males, small amounts of estrogen are produced by the testes. So our testes do produce estrogen. The enzyme aromatase, found in various tissues including the testes, converts a small portion of testosterone into estrogen, while estrogen levels in males are lower than in females. It plays a role in various physiological processes, including bone health and reproductive function. Progesterone is typically at much lower levels in males, and it is produced in small amounts by the testes and adrenal glands. The function in males is not well understood as in females, but it is thought to contribute to some aspects of male reproductive function. Females do produce testosterone. The ovaries are primary source for testosterone in females, and adrenal glands, which sit on top of kidneys, also produce small amounts of testosterone. After menopause, when ovarian function declines in females, some estrogen is still produced by peripheral tissues through a process called aromatization. So, yes. Okay. Upon ovulation, the oocyte is released into the fallopian tube, I believe. So, we answered D, but it is C. Oh, it's in the abdominal cavity. That was kind of my guess too, but let's see. The subtle point about ovulation is missed by most students and remains hard to believe until the organs are examined in anatomy class in medical school. The ruptured ovarian follicle releases an oocyte into the abdominal cavity. So we had the picture here. Let's look at it again. We have the ruptured follicle here during ovulation that releases the oocyte. Is this the primary oocyte or the secondary? It is the secondary oocyte, as we had it here, right? It's called the secondary oocyte at ovulation. The ruptured ovarian follicle releases an oocyte into the abdominal cavity, close to the entrance of the fallopian tube. With the aid of beating cilia, the oocyte is drawn into the fallopian tube, through which it travels until it reaches the uterus. If it is fertilized in the fallopian tube, it will implant in the uterine wall. If fertilization does not occur, it will be expelled along with the uterine lining during menstruation. Okay, nice. Let's see if we can find an image of abdominal cavity. So, okay, this is not helpful. Let's go to images. This is kind of what I like to see, but that is weird. That is for a mouse or something. It's not for a human being. So the abdominal cavity is on the upper section. We have the pelvic cavity. We have the abdominal pelvic cavity both together. Let's see what we can find for yeah, so and fallopian tube. Okay, So we have, okay, let's go from the vagina. So we have the vagina here. We have the cervix of uterus. We have the myometrium. We have the uterosacral ligament. We have body of uterus. We have mesometrium. We have proper ovarian ligament. We have the broad ligament of uterus. We have the ovary. We have the mesosalpinx. Mesosalpinx. We have the isthmus of uterine tube. We have the vesicular appendage of epophoron. We have the infundibulum of uterine tube. We have the epophoron. We have the ampulla of uterine tube. And then we have the fundus. Now we have the external os of uterus. We have internal os of uterus. We have the vaginal fornix. We have the vaginal fornix. We have the isthmus of uterus. Endometrium is here. So we have the ureter. We have the uterine ostium of uterine tube. We have meso-ovarium. We have corpus albicans. We have corpus luteum. We have tertiary ovarian follicle. We have fimbriae of uterine tube. And uterine tube. I mean, I came here for the fallopian tube. Is this the same as uterine tube? Is the fallopian tube the same as uterine tube? Yes. So there's the same. The fallopian tube, the uterine tube is on this thing. And you can see that the beginning of it, the opening, is where we have the abdominal cavity, right? Ruptured ovarian follicle releases oocyte into the abdominal cavity close to the entrance of fallopian tube. With the aid of beating cilia, the oocyte is drawn into the fallopian tube, through which it travels until it reaches the uterus. So the uterus, where's the uterus? We had it here. This is the cervix of uterus. Oh, so it goes from the outside to the inside, right? The ureter. These are for like urine, right? Isthmus of uterus. If it is fertilized in the fallopian tube, it will implant in the uterine wall. If fertilization does not occur, it will be expelled along with the uterine lining during menstruation. Okay. So the direction of the oocyte when not fertilized is from the ovaries to the fallopian tube to the uterus to the endometrium, and if fertilized, it is done so in the fallopian tube. Yes, that's correct. So we have the ovulation. The mature egg is released from one of the ovaries. The egg is then captured by the fimbriae, which are finger-like projections at the end of the fallopian tube. The egg travels through the fallopian tube towards the uterus. The fallopian tube is the site where fertilization typically occurs if sperm is present. The journey through the tube takes several days. If sperm is present in the fallopian tube and successfully fertilizes the egg, the resulting fertilized egg, now called a zygote, and successfully fertilizes the egg, the resulting fertilized egg, now called a zygote. So zygote is the fertilized egg. It begins to divide. Fertilization usually occurs in the ampulla, which is the middle portion of the fallopian tube. The early stage embryo continues to divide as it travels through the fallopian tube. By the time it reaches the uterus, the embryo is a blastocyte. So the early stage embryo's journey to the uterus. So fertilization usually occurs in the ampulla, which is the middle portion of the fallopian tube. The early stage embryo continues to divide as it travels through the fallopian tube. By the time it reaches the uterus, again, we have uterus. By the time it comes to the middle, the embryo is a blastocyst. Excuse me. So, yeah, we don't have the stuff. Oh, here it is. So we have all these things on chapter 3. Okay, cool. So chapter 3 is basically embryogenesis and development. Yeah, you can see here all the things about the uterine cavities, stuff like that. Archentron leads to blastopore. Then we have nervous system, skin, hair, nails, mouth, anus, tractoderm, ectoderm. Okay. Okay. If conditions are favorable, the blastocyst will implant itself in the endometrial lining of the uterus. This marks the beginning of pregnancy, and we know that the endometrial lining is here. Okay. Let's continue. we have then question 7. So upon ovulation, the oocyte is released into the abdominal cavity. So we must have a takeaway here. Oocyte into abdominal cavity. Secondary oocyte, right? This abdominal cavity is the entrance of the uterus, right? Entrance of fallopian tube. And then what happens after this? Does the oocyte go to the middle section, to the uterus? So with the aid of so then cilia help oocyte to enter fallopian tube. Then there, if fertilized, it is through which it travels until it reaches the uterus. If it's fertilized, it will implant in uterine wall. So and uterus, but the fertilization happens in fallopian tube. So yeah, and then uterus. If fertilized, implanted in endometrial wall, or not fertilized, or expelled via menstruation with endometrial lining. Most of the time it's going to be like that, right? It's expelled with you don't have babies every here and then. Okay, next question. Cancer cells are cells in which mitosis occurs continuously without regard to quality or quantity of the cells produced. Chemotherapies attack rapidly dividing cells. At which point in the cell cycle could chemotherapy effectively prevent cancer cell division? So we know the S stage is the growth stage, basically the the DNA replication. The prophase and metaphase, first and second stage of the mitosis. So if you want cell division stopped, I would honestly attack all of them. 1, 2, and 3. I know I answered 1 and 2, I think, or 1 only. Let's see, 7 is D. Yeah, so the question is asking us to determine at which points in the cell cycle we prevent or at least lower the number of cells undergoing mitosis. One idea would be to prevent DNA synthesis during S stage, Without DNA being replicated, two variable daughters could not be formed. Our ideas would be preventing the mitotic cycle from forming alternating prophase by preventing spindle apparatus formation, preventing nuclear membrane from dissolving or interfering with other processes during the phase. Similar treatment that would act on cells in the metaphase stage of the cycle would also interfere with the mitotic cycle. Therefore, any of the three solutions presented would be a viable option. Which of the following incorrectly pairs the structure of male? Yeah, so for 8, I chose B. I was correct. So, prostate gland surrounded by muscle to raise and lower the testes. That is the job of the detrusor muscle, right? So we have the prostate gland, along with the seminal vesicles and bulbourethral gland, secretes seminal fluid. So we have the prostate gland, seminal vesicles, and bulbourethral gland. They do this seminal fluid, which combines with sperm to produce semen. It is the cremaster muscle which surrounds the testes that raises and lowers the testes in response to changes in temperature. Interesting. It's the cremaster muscle. So we have, let's write cremaster muscle. The detrusor muscle was for the urine secretion, right? Excretion. You know, like forcing our bladder, I believe, to squeeze the urine out of it. Cremaster muscle, raise, lower, testes due to temperature. So we can put this here. Okay. So what was the 7-up again? So S is the seminiferous tubules. E is the epididymis. V is vas deferens. Next E is for ejaculatory duct, and the U is for urethra, and P is for penis. Urethra is, yeah. Okay. So the R1 vas deferens tube connecting the epididymis to the ejaculatory duct. We have vas deferens to raising and lowering testes. Does vas deferens lower, raise testes? Maybe cremaster muscle is more. Does not lower raise testes. The vas deferens is a duct that transports sperm from the test to the urethra, connecting epididymis to the ejaculatory duct. It transfers. Where is that? Transports sperm from the testes to the urethra, which is part of the male reproductive system. It plays a crucial role in the passage of sperm during ejaculation. The testes, on the other hand, are suspended outside the abdominal cavity in the scrotum. So it's between vas deferens connecting epididymis to ejaculatory duct. What's the Cooper's glands? produce a fluid to clear traces of urine in the urethra. Testes, on the other hand, are suspended outside the abdominal cavity. Position of testes in the scrotum is influenced by the contraction of muscles in spermatic cord. These muscles respond to, and the cremaster muscle, to temperature changes and other factors to regulate the muscles. Help to maintain optimal temperature for sperm production. Scrotum. The testes need to be kept slightly cooler than the body's core temperature for the efficient production of sperm. When it's too warm, the muscles in the spermatic cord relax, allowing the testes to hang lower, away from the warmth of the body. Conversely, when it's colder. So, what is the Cooper's gland? Bulbourethral gland. So, bulbourethral gland. So this goes for this. aka Cooper's gland. Probably name of the scientist. Is there a picture of the bulbourethral gland? Here it is. By bulbourethral gland, bulb of penis, urethra, bulbourethral gland, and this is our bladder. So the Cooper's gland, it makes viscous fluid to clean out urethra, just as we thought. Produce a fluid to clear traces of urine. Okay. What is the last point in the meiotic cycle in which the cell has a diploid number of chromosomes? So as we got to know, it must be, so they are rest in the prophase 1. So, but this is different. Where is interphase and what is interkinesis? So interphase is this, right? And interkinesis is here. So interphase is the beginning. It should be interkinesis, right? Where we have the haploid or telophase 1. I think I will go with telophase 1, perhaps. Is this 9? The first meiotic division, reductional division, pulls homologous chromosomes to opposite poles of the cell during anaphase 1. Near the end of telophase 1, cytokinesis occurs, resulting in 2 haploid daughter cells. Thus, during interkinesis in anaphase 2, the daughter cells are already haploid. In eliminating C and D, interkinesis in telophase 2, the cell is diploid during interphase, but remains diploid up until the end of telophase 1. So 9 is B. Last point. It's telophase 1. Okay. Which of the following does not likely contribute to genetic variability? So we have question 10. We answered correctly. It's D. Which of the following does not likely contribute to genetic variability? Replication of the DNA during S stage. So let's just finish these quickly. Which of the following does not likely contribute to genetic variability? The safest way to answer this question is to go through each answer choice and eliminate the ones. The random fertilization of an egg by a sperm, the random segregation of homologous chromosomes during anaphase, and crossing over between homologous chromosomes during prophase 1 all contribute to genetic variability during sexual reproduction because they result in novel combinations of genetic material. S stage, D, should not cause increased genetic variability. The DNA should be copied precisely without error, and meaning that both strands of DNA should be identical, right? It's a replication, it's not genetic variability. 11, we answered correctly. Which of the following statements correctly identifies the key difference between mitosis and meiosis? During metaphase of mitosis, centromeres they are present directly on the metaphase plate. During metaphase, there are no centromeres on the metaphase. The key difference between mitosis and primarily during meiosis 1. Of note, synapses and crossing over occur during prophase 1. Homologous chromosomes are separated during so this is called synapses and crossing over. We have it here, right? Crossing over in meiosis. What is crossing over in meiosis? Prophase 1. Genetic recombination, crucial event that occurs during prophase 1. Cell division, process that leads to formation of gametes in sexually reproducing organisms. Prophase 1 is the first stage of meiosis 1. Crossing over takes place. During crossing over, homologous chromosomes exchange genetic material, specifically segments of chromatids. Homologous chromosomes are pairs of chromosomes that have the same genes but may have different alleles. Here's a step-by-step. They pair up. In prophase 1, homologous chromosomes align next to each other, forming a structure called a tetrad. Tetrads consist of 2 homologous chromosomes, each composed of 2 sister chromatids. During the alignment of homologous chromosomes, sections of chromatids from one chromosome can break and exchange places with the corresponding sections of the other chromosome in the tetrad. This exchange of genetic material results in crossover or recombination. So recombination or crossover. So we have aka recombination. Resulting tetrad. Cool. Individual who is phenotypically female. I answered 45X for this one. So we got, I think, C, right? Let's see. Oh, which of the following is true regarding prophase? The nucleoli disappear. That's what I answered. So let's go here. In prophase, the chromatin condenses into chromosomes, the spindle apparatus forms, and the nucleoli and nuclear membrane disappear. A describes anaphase, where B and C describe telophase. So chromosomes separate and move to opposite poles of the cell. That's for separate and move to opposite poles. This is for anaphase, the separation. Spindle apparatus disappears. This is for telophase. Chromosomes uncoil for the chromosomes uncoil, so decondense. That's for telophase. So telo, and N. The condensation happens in prophase. And then the next question, non-disjunction refers to the incorrect segregation of homologous chromosomes during anaphase 1 or of sister chromatids during anaphase 2. In either case, one daughter cell ends up with 2 copies of related genetic material while the other receives 0. Immediately, this should eliminate A and B, which show a normal complement of chromosomes 46. So where do we see non-disjunction? This is one of the homologous chromosomes gets, so one daughter cell ends up with 2 copies, so not one chromatid but 2 chromatids, but the other one is with 0 copies. An individual who has only one recessive disease-carrying allele and yet still expresses the disease likely does not have a dominant allele for the given trait. This is seen in males who are hemizygous for many X-linked genes and can also be seen in women with Turner syndrome 45X who have only one X chromosome. Turner syndrome. Ooh, 45X Turner syndrome. Take away Turner syndrome 45X. During which, like, remember Tina Turner? She was 45 probably when she was singing the amazing songs she was singing. During which phase of the menstrual cycle does progesterone concentration peak? Come on, that is in the luteal phase. So, sorry. Oh, I'm sorry. Yeah. Progesterone peaks during the luteal phase as it supports the endometrium for potential implementation of a blastula. Progesterone levels are relatively low during the follicular phase and ovulation. Withdrawal of progesterone actually causes menses. How do you say this?
Menses.
Menses. What is what is menses? Menstrual phase of the menstrual cycle in females. So we have a menstrual follicular phase in luteal. Menses is this bar. So it's the first 7 days. Is a term commonly used to describe a regular monthly shedding of the uterine lining, which is expelled from the body through the vaginal. Menstruation is a natural part of the menstrual cycle and is often colloquially referred to as woman's period. So the period is basically the same as menses. Menstrual cycle. The menstrual cycle is a series of physiological changes that occur in the female reproductive system, typically lasting 28 days. Although variations are common, the cycle involves the preparation of the uterus for a potential pregnancy. Menses phase. The menses phase is the first part of the menstrual cycle. It begins from usually lasts 3 to 7 days. When the uterine lining known as the endometrium is shed, so you have your period. Cool. So the peak of progesterone is in the luteal phase. And how about the estrogen? That is in the right at the ovulation. During the first trimester of pregnancy, the corpus luteum is preserved by human chorionic gonadotropin HCG, hence progesterone secretion by the corpus luteum is maintained during the first trimester. This eliminates wait. Progesterone levels are relatively low during follicular phase, which is the first phase, not luteal, follicular, and ovulation. Withdrawal of progesterone actually causes menses. So what causes menses is the withdrawal of progesterone. Withdrawal of progesterone causes menses. Okay, last one, which of the following would not be seen during pregnancy? Okay, what is HCG? In the first trimester of pregnancy, the corpus luteum is preserved by human chorionic gonadotropin HCG. Hence, progesterone secretion by the corpus luteum is maintained during the first trimester. High levels of HCG in the first trimester. We eliminated because progesterone secretion by corpus luteum is maintained during the first trimester. The corpus luteum is preserved by human progesterone. During the second trimester, HCG levels decline, but so we do see high levels of HCG in the first trimester, high levels of progesterone throughout the pregnancy. But during the second trimester, the HCG levels drop, but progesterone levels rise because the hormone is now secreted by the placenta itself. High levels of progesterone and estrogen inhibit GnRH. Low levels of FSH in the first trimester. High levels of progesterone and estrogen inhibit GnRH secretion, gonadotropin releasing hormone, thus preventing FSH and LH secretion and the onset of a new menstrual cycle, and validates D, thus preventing high levels of GnRH throughout the pregnancy. Inhibit GnRH, thus preventing FSH and LH secretion and the onset of a new menstrual cycle. Okay, I'll take a quick break and be right back.